I would like to thank the organizers for inviting me to discuss feeding the gut microbiome in clinical practice today. I would like you to be aware of my disclosures. And so the aims of my presentation today is to discuss an overview of dietary patterns that may affect the gut microbiome. From the other two talks today, you will hear that there are many other factors that can affect gut microbiome, such as where we live and our stress levels. So diet is just one component that we will be addressing in my presentation. We will discuss the overview of dietary factors, so other than dietary patterns that may affect the gut microbiome, the role of pre, pro, and symbiotics with a particular focus on probiotics, and then practical information. So I was interested to learn that just calorie restriction in itself can affect the gut microbiome. As you will see on this slide, this study was conducted in 15 obese adults, all with non-alcoholic fatty liver disease. There were some species-specific differences seen in the group that was on the lower calorie diet versus the ones on the normal calorie diet. And we also saw a change in butyrate production. The magic word, which you will hear a lot about today. Butyrate, of course, is one of the short-chain fatty acids. And the authors then concluded to say that this change in the microbiome and increase in butyrate production could have an effect on the possible prevention of non-communicable diseases such as NAFLD. Okay, then let's talk about diet diversity in the microbiome. We have some really nice data indicating that in infancy, healthy diet diversity is associated with increased microbiome diversity measured by the Shannon index. And we also have data, which you will hear in one of the other talks about as well, that a healthy diet pattern consisting of fish, yogurt, and a range of vegetables is associated with, once again, increased short-chain fatty acid, particularly butyrate production. Looking further at diet diversity in the infant microbiome, and sometimes it is really fascinating how little data we have about infant nutrition. And so they really, other than the data I showed from Caroline Reday's team on the previous slide, no data looking just at simple diet diversity in infancy and how that affects gut microbiome diversity. But if we look at this paper by Lawson et al., where they looked at microbiome diversity at 6 months, when infants started to introduce solid foods in their diet up to 12 months of age, which is by the time they were more or less on a family-based diet, they found that the transition to family foods with a higher protein and fiber content correlated with an increase in gut microbiome diversity. So currently in the United States, the American Academy of Pediatric still recommend that we should wait about 3 to 5 days between introducing new foods. But if you start introduction at 6 months, you're only going to get to about 35 foods by 12 months of age. And these foods also need to include fat-containing foods and protein-containing foods. So that really makes it very difficult to get to a very diverse diet by 12 months of age. And this is perhaps guidance that needs to be revisited again. So then fat, the majority of studies that we have looking at the Western diet versus microbial outcomes such as diversity, function, or species-specific differences look at the Western diet in animal models, particularly mouse models. And there is the question about how relevant these mouse model studies and the diets they feed the mice are in terms of human diets. So the mice studies often contain a lot of lard, refined sugar, which in the human world would be equivalent to a diet predominantly consisting of pork rinds, ribs, and coke. And most humans, even if they have a diet that needs some improvement, do not basically eat all these foods all the time. So there's some questions about how to translate the mouse model data into human data. But there you can see, looking at the mouse model data, clear differences we see in the mice on a Western-based diet versus those on a normal mouse chow diet. Then when we look at human data now, and we focus again on infants, getting back to the study by Lawson et al., we can see that as fat intake increased, so at this point, the children had predominantly purified fruit, or pureed, I should say, pureed fruit and vegetables in their diet. They had a much higher microbial diversity, but when they got to 12 months of age, with about 60% fat in their diet, mimicking what the rest of the family were eating, we saw a reduction in gut microbiome diversity. So clearly the practical message is, yes, we do want babies to eat lots of fruit and vegetables. We want them to introduce fats and protein foods alongside the fruit and vegetables in their diet, but overconsumption of fatty foods, particularly in infancy, may not be that beneficial to gut microbiome function. Then often I get asked, does the fat matter? I can show you many slides with lots of information looking at different kinds of fat and how they affect the gut microbiome. But I think it is safe for clinical practice to conclude at this point in time that healthy eating guidance prevail. We have to watch the fat. We do want to focus on monounsaturated fats such as olive oil in the diet, but it doesn't mean that children can never be allowed to have any saturated fats or any polyunsaturated fats. It's all about balance at the end of the day. Then moving on to fiber, I'm sure all of you in this audience have at least read one paper in your life about the role of fiber intake on the microbiome. And this is a beautiful study comparing the gut microbiome diversity or richness, biodiversity, and also species-specific differences between African children and children based in the EU. So clearly, you know, fiber intake was different between the two groups, explaining the differences we see in the gut microbiome diversity. But as you will hear from the other talks, we really also should take into account that the living arrangements of children in Africa and that the community of humans that they come in contact with will be very different from the communities that children live in Europe, as well as the other humans that they come in contact with. The American Gut Study, I really do like the data from the American Gut Study, and they've shown that adults who eat more than 30 different plant-based foods a week have a very different gut microbiome structure and function than adults who eat only 10 plant-based foods. Not all the differences were statistically significant, but I do think we have enough evidence to say that we really should increase the number and diversity of plant-based foods, definitely in adults, but I also translate that into the clinical advice I give to the families I work with infants and young children. Fermented foods is another buzzword alongside butyrate. There's many different studies looking at how fermented food can affect gut microbial function. There's particularly many studies that actually measured the different types of bacteria in all these different fermented foods. And so we know that amongst this list of about 10 different fermented foods, hundreds of different species differences have been seen in terms of bacteria content of these foods. But when we drill down again to clinical practice, there's only really one study that used fermented foods in a trial and looked at differences in gut microbiome structure and function. And this was a study that gave kefir for 4 weeks to adults with ulcerative colitis, and they found higher abundance of lactobacillus in particular in the group that received the kefir. So really getting back to the practical information, because this is what it's about. What can we tell our patients? We want infants and young children to have a diverse diet across all the food groups, not just fruit diversity, not just vegetable. We want a diverse diet in total. We want to focus on plant-based food, even though they're not the only foods that we're allowing in the diet, trying to get to about 30 different plant-based foods in the diet a week. And these portions can be very small for young infants. We want to watch the fat, but we want to allow the fat. We want to make sure there's adequate amount of protein in the diet. We want to focus on fermented food. For some families, that may only mean daily intake of yogurt. For other families, it may mean eating a variety of different fermented foods. And then normally, just as the families leave the door, they turn around and say, but is there not some supplement that I can give the child to benefit their microbiome? And in this point in time, we probably have the best data from the studies using the bacteria referred to as Lactobacillus GG. And we can see here in a study conducted in Italy that the children had cow's milk allergy, but the children who received the supplement with the added Lactobacillus GG also had less atopic manifestations. So outside of the fact that the children developed tolerance to cow's milk allergy, or tolerance to cow's milk quicker, that they had less symptoms from their cow's milk allergy, they also had less other atopic manifestations, which is really what we want at the end of the day. So with this, I'm just going to summarize. A healthy, diverse diet is important. We want a variety of foods in children's diet. We do want to allow some protein and fat in their diet. We want to give them practical written advice. That's what dietitians are really good at. And we want to give them information on pre, pro, and symbiotic intake. We have the best data about probiotics at this point in time. And I think it's an exciting time to learn about this field more and more every day. So thank you for your time. And I look forward to further discussions during question and answer.